Hello, I'm Sergio Giralt, and I'm speaking to you from here, Boston, where we are at the Myeloma 2015 meeting. I'm joined by Dr. Faith Davies and Dr. Shaji Kumar. We just finished a very interesting session regarding novel targets, particularly looking at kinases. Faith, you spoke to us about MEK inhibitors, and it seems like not only are there drugs available, but there is some activity in myeloma. What do you feel the future of this area is?
So we were able to show our experience of MEK inhibitors in over 50 patients. And we've known for many years that the RAS pathway is activated in myeloma patients. And we chose to concentrate on those patients that have got mutations in the RAS pathway. And we could show that with single agent activity, we had responses of PR or better in 15% of patients. Or if we looked at MR or better, then we had responses of 35% or more. And I think that latter measurement is really important because the patients in our study had at least 5 lines of therapy previously. They had high-risk features, so they were a very group of patients with very, very poor prognostic features. And despite that, we showed excellent activity both as a single agent, but also in combination with other agents such as bortezomib, lenalidomide, and the chemotherapy agents. As I say, we chose to concentrate on those cases that were mutated, but the biology data suggests there's actually many other patients that have activation of that pathway, which could also benefit from MEK inhibitors.
Despite not having the mutation necessarily.
Despite not having the mutation, yes. So we know that patients may have activation demonstrated by phosphorylation on immunohistochemistry, but also a number of patients may have an MMSET or high MAF levels, and those patients also seem to have activation of the RAS-RAF kinase, MAP kinase pathway.
And this has been a pathway that had not been explored until today, correct?
Yeah, no. So it's really quite exciting, and one of the nice things is that the drug we've been using is actually a drug that is FDA approved for melanoma. So it is actually very close to the clinic and very achievable to actually get it for patients.
And other better drugs coming down the road potentially, right?
Yes, so there's other MEK inhibitors that are coming down the road which maybe have improved safety profile. But also you can actually target that pathway at a number of different levels. And so it may well be that we could choose a different node on the pathway and get better responses, or indeed combine drugs together and overcome resistance and get better responses.
Interesting times. Shaji, you talked to us about also a new pathway that's being explored in myeloma, the PIM kinase pathway, correct?
Right.
Tell a little bit about that. You're just finishing the phase 1, right? You're doing the expansion now.
That is correct. So the PIM kinases are something that is, I think, relatively new in terms of its contribution to the tumor, particularly hematological cancers. So it seems like it has a very critical role in maintaining the cell survival, and it interacts with some of the cell survival signaling pathways in different cancers, especially in leukemias and myelomas. So this was the first in human trial of this drug called LGH447 or PIM447, as it is known right now. And the study, almost 60 patients were studied in this trial, starting from a very low dose of about 70 milligrams all the way up to 700 milligrams. And what was interesting was we did see actually responses in some of these patients. There was one patient who actually had a very good partial response. And again, considering that we took a patient with a median of 4 prior lines of therapy, so fairly heavily pretreated patients, vast majority of them had a previous transplant or had been exposed to IMiDs and proteasome inhibitors. We also saw about 1 in 5 of these patients actually had at least a minimal response or better, and a fair number of patients who had stable diseases. And the duration of response was pretty good. I think the ones who had the response, there was at least one patient who went all the way up to 6 months of continued response. So I think the early signal clearly appears that there is activity, or at least some role for this pathway to be inhibited or targeted in this disease. Now, we did see some side effects. We certainly saw some platelet count problems. The thrombocytopenia, again, was mostly manageable with dose modifications, but the vast majority of the patients who went off the study was because the myeloma started progressing.
And here, both you and I think our European colleagues are thinking combination therapy, that although because of drug-drug interactions, the drug cannot be combined with bortezomib, but it seemed to be safely combined with any of the IMiDs and with carfilzomib. Am I correct?
That's right. So at least the preclinical work suggests that it may be some benefit in combining these classes of drugs, and there's clear synergy. I think the clinical trials are currently being started looking at these combinations. Not only can it be combined with some of these drugs that are currently in the clinic, at least from a mechanism standpoint, it seems like you could combine it with other PI3 kinase AKT pathway inhibitor, and one of those clinical trials combining the PIM447 with another Novartis drug, BLY, is currently ongoing.
I think we can all say, even for the patients, that this has been an excellent Myeloma 2015 meeting, that we're now, we've discovered new pathways that we can look, sometimes for very targeted patients, sometimes for a large population of patients, that continued participation in clinical trials will actually move the field a lot quicker. And now we're coming for the very interesting question of cellular therapies and immunotherapies that we get to see now. Thank you very much, both of you, and congratulations for great work and great presentation.
Thanks,
Thank
Sergio.
you.
From Boston, Sergio Giralt.